DNA repair. In this chapter, we are going to learn necessity of DNA repair, mistakes in DNA, agents causing mistakes and damage in DNA, biochemical mechanism of DNA repair, mismatch repair, thymine dimer repair, photoreactivation, then base excision repair, recombination and repair, SOS repair or error-prone repair, extensive damage repair by homologous recombinations. Necessity of DNA repair. DNA molecules are most vital for the cell and for the survival of individual organisms. But DNA molecules are quite fragile and are constantly subjected to radiations and intracellular physical and chemical stresses. Oxygen, UV light, X-rays, alkylating agents, and free radicals all can cause damage and introduce changes in nucleotide sequence. These changes can result from breaks in polynucleotide strands, chemical modifications of nitrogenous bases in the strands, and breaks in polynucleotide strands. Incorporation of mismatched bases during replication. The commonly occurring changes in DNA can be due to formation of pyrimidine dimers, then alkylation or deamination of nitrogenous bases, both spontaneous and induced changes in DNA. If left uncorrected, they may result in mutations that disrupt the function of essential enzymes or structural proteins and pose a significant risk to the existence of cell. For correcting such occasional mistakes in DNA structure, several efficient correcting mechanisms have evolved in order to protect organisms against potential deleterious effects of mutations. Mistakes in DNA. Types of mistakes. The various mistakes that are often introduced in the nucleotide sequence in DNA replication can be classified into following categories. First, single strand breaks in DNA, double strand breaks in DNA, alteration of bases such as intrastrand dimer formed by two adjacent pyrimidines, thymine dimer by UV radiation. Mistakes. First, insertion of incorrect base in one strand. Insertion of incorrect base in one strand. That causes replication error. Insertion of incorrect base in one strand causes replication error. For example, replacement of purine by pyrimidine or vice versa. That will cause replication error. Then spontaneous deamination of pyrimidine C to U, spontaneous deamination of purine A to hypoxanthine, etc. Then alternation of bases, introduction by ionizing radiations and UV rays. They lead to the formation of thymine dimers. Loss of bases, introduced by alkylating agents that cause depurination by breaking of N-glycosidic bond. Then single strand breaks caused due to the breakage of phosphodiester bonds by chemicals, peroxides, sulfhydryl containing compounds, by losing radiation, and by free radicals in water. Then double strand breaks are single strand breaks introduced in separate strands of DNA at the same or in different levels. Cross linking of the two chains of the helix are antibody mediated covalent linkages between a base of the one strand and an opposite stranded complementary DNA strand. Agents causing mistake and damage. The various agents causing damage to DNA or introducing mistakes in nucleotide sequences can be placed under following categories like radiation, ionizing radiation, for example, X-rays, partly occurring cosmic rays, electron beams, gamma rays, and radiation beams of various kinds used in radiothermy are associated with breaks in polynucleotide strands of DNA. The strands breaks are caused by the action of secondary electrons produced by beta particle or the X-ray photon, or by the production of free radicals in water that can attack the phosphodiester bonds. Chemicals. Strong chemicals like alkylating agents such as ethane sulfonate or MES, ethyl ethane sulfonate ES, and nitroguanidine react primarily with guanine by adding an alkyl group to N7 of purine ring. This weakens the N-glycosidic bond and causes depurination. Depurination. Depurination occurs by the breakage of N-glycosidic bond of a purine nucleotide. The rate of spontaneous depurination is about 1 purine removed over 300 purines per day. Afloxacin, benzopyrene, nitrosamines, nitrosorine, and cisplatin. Cisplatin are some of the chemical agents or drugs which cause damage to DNA and introduce gene mutations. Biochemical mechanism of DNA repair. Biochemical mechanism of DNA repair. Cells possess a variety of mechanisms to repair the damages in DNA structure caused by various mutagenic agents. There are repair of damage caused by incorrect base insertion or mismatch repair. During DNA replication, both the enzyme polymerase 1 and polymerase 3 occasionally incorporate wrong or incorrect base in the daughter strand under synthesis. A wrong base fails to form a hydrogen bond with the template base in the parent or template strand. Such errors are usually corrected by the editing function of these enzymes. The errors missed by the editing function of these enzymes are corrected by the second correcting system called the mismatch repair. In mismatch repair, a pair of non-hydrogen bonding bases is recognized as incorrect and a polynucleotide segment with unmatched pair is excised, thereby removing the mismatched pair. This gap is filled by polymerase 1. This mismatch repair system also counteracts the mutagenic effects of tautomeric bases. Such molecules in the tautomeric form are misincorporated into DNA because tautomeric A the pair with C, tautomeric G pairs with T. Such pairs are correctly hydrogen bonded to template strand and non-recognized by editing enzyme. But when tautomeric base returns to its original form, to form a mismatched base pair is corrected by mismatch repair system. Mismatch repair system. The mismatch repair system requires a complex of these three proteins, MUTS, MUTH, and MUTL. The protein MUTS recognizes the binds to the mismatched base. MUTH binds to DNA at the hemimethylated GATC site and cleaves the unmethylated newly synthesized strand. MUTL binds to MUTS and MUTH and helps in final repair steps. Extension mismatch repair genes. Mismatch repair is a highly conserved mechanism. It functions in similar fashion in bacteria as well as in eukaryotes. It is believed that families of eukaryotic genes homologous to MUTS, MUTH, and MUTL chose before the evolutionary divergence of animals, plants, and fungi groups. Repair of thymidine dimers. This can be discussed under following headings. Formation of pyrimidine dimers. Radiations produce intrastrand photodimers between two adjacent pyrimidine bases of same strand. The dimers result from the formation of cyclobutyl ring by the joining of fifth and sixth carbon atoms of the two pyrimidine rings. Such cyclobutyl pyrimidine dimers are most common photodimers and thymidine dimers are commonest of all. Effect of dimers. The presence of thymine dimer or any other pyrimidine dimer causes distortion of DNA helix because the two thymines are pulled closer. This produces a kink in the polynucleotide strand in which dimer is formed. The kink causes an inhibition of the advancement of replication fork and this interfere with DNA replication. Mechanism of dimer repair. Effect of dimers. The presence of thymine dimers or any other pyrimidine dimer causes distortion of DNA helix because of two thymines are pulled closer. This produces a kink. We learned about this. Mechanism of dimer repair. There are four major pathways for the repair or damage repair in DNA caused by thymine dimers. These are separated into two categories: photoreactivation or light-induced repair, light-independent repair, excision repair, recombination repair, and SOS repair. These are different mechanisms. So, photoreactivation or light-induced repair is some sort of different method. Photoreactivation or light-induced repair. Photoreactivation or light-induced repair. Photoreactivation is light-induced enzymatic cleavage of pyrimidine dimer, thymidine dimer. It is catalyzed by enzymes called photolysis or PR enzymes. They absorb radiant energy of wavelength 300 to 600 nm and split the cyclobutyl ring between two pyrimidine molecules of the dimer. As a result, the cyclobutyl thymidine dimers are converted to pyrimidine monomers. Thymidine dimers are converted to monomers. Excision repair or dark repair. The excision repair mechanism is a multi-step enzymatic process that can remove and replace individual nitrogenous bases, base excision repair, and also the longer nucleotide chains, nucleotide repair. This repair mechanism does not require sunlight, so it is also called dark repair. Base excision repair includes enzymatic removal of chemically modified bases such as uracil 5-methyl cytosine or 3-methyl adenine, followed by removal of the nucleotide. The gap thus created is filled by a complementary base pair. It is carried out in the following steps with the aid of several enzymes. The enzyme DNA glycosylase removes the chemically modified base, causing upurination or apyrimidation. The enzyme endonuclease cleaves the phosphodiester bond, the sugar phosphate backbone at the aspirinic or apyrimidinic site, forming an egg. Enzyme deoxyribose phosphodiesterase removes the deoxyribose phosphate molecule. The repair polymerase DNA polymerase beta adds a single nucleotide to the 3' end of the naked strand at the gap. Enzyme DNA ligase seals the nick by joining the two parts of the polynucleotide strand. Nucleotide excision repair operates to remove pyrimidine dimer or thymidine dimer and fills the gap by complementary nucleotides. Require the entire DNA replication machinery. The enzyme repair endonuclease recognizes the distortion of the polynucleotide strand produced by a thymidine dimer. It cleaves the sugar phosphate backbone of the DNA strand on both the sides of the kink. This exposes 3' OH group at the 5' incision site. The oligonucleotide consists of about 30 nucleotides removed by DNA helicase. The resulting gap is formed repaired by DNA polymerase delta or epsilon and is closed by DNA ligase. Next, recombination repair. In recombination repair, the undamaged strand of parental DNA is copied during replication to produce a normal daughter molecule. During DNA replication, polymerase III is stalled at the site of thymine dimer. The enzyme passes on to the dimer block and initiates chain growth beyond the block. Therefore, daughter strands have large gaps, one for each thymidine dimer. Complete normal double-strand break molecules are formed from such fragmented and kinked DNA molecules by sister strand exchange mechanism. In single strand exchange, the gap produced in the daughter strand is filled by recombination with the undamaged parent strand due to its exchange. A gap is formed in the previously intact parent strand. This is filled by the action of DNA polymerase and ligase enzyme. The pyrimidine dimer is then later on removed by excision repair to produce two intact normal double-stranded DNA molecules. Pyrimidine dimer replication is blocked by pyrimidine dimer polymerase reinitiated downstream from damaged site. A gap is present in daughter strand opposite dimer site. Recombination leaves gap in previously intact parent strand. Daughter strand with recombination gap in parent strand is filled. That is post-replication repair by recombination. Replicating DNA molecule with a pyrimidine dimer, DNA polymerase bypasses the dimer and starts post-dimer synthesis of new strand. The resultant gap in the newly synthesized DNA strand is repaired by combination with undamaged parent strand. Enzyme DNA polymerase and DNA ligase fill the resultant gap in the previously intact parent strand. Next, SOS repair or error-prone repair. SOS repair is a bypass repair system that allows polymerization to proceed across a dimer despite the restoration of the helix. During replication, the DNA polymerase complex bypasses the damaged site in the template strand and continues DNA synthesis. It inserts adenine nucleotides at the gap regardless of original DNA sequence. Thus, this repair system is potentially mutagenic. In SOS repair, REC-A protein binds tightly to the dimer strand. When DNA polymer 3 encounters the dimer, REC-A interacts with the epsilon unit of polymerase 3, which is responsible for DNA editing. As a result, epsilon unit failed to proofread and edit the insertion of mismatched base and allows replication to proceed to overlooking the insertion of wrong base. In E. coli proteins, UMUC and UMUD bind to DNA polymerase and alter the stringency of nucleotide incorporation. Adenines are inserted into the newly synthesized strand at the junction lesion site, irrespective of base sequence in the parent template strand. If the damaged site is thymidine dimer, the newly synthesized DNA is not mutated. If the damaged site has cytosine dimer, the newly synthesized strand is mutated. A SOS repair system operates only under potentially lethal conditions caused by extensive UV radiation. Therefore, a SOS repair system works on the principle that mutation is better than no survival at all. This repair is also called error-prone repair or translation replication. Extension role of genes MUMC and MUMD in SOS repair. The proteins produced by UMUC and UMUD gene family are necessary for dimer bypass. They perform following functions. They help in tight binding of REC-A protein in the small decoction caused by pyrimidine dimer. They help in binding of RNA polymerase 3 to distorted region. They help polymerase 3 to bypass the damaged site in template DNA and move forward in the direction of advancing replication. Fork genes homologous to UMUC and UMUD are found in almost all animals and plants. Extensive damage repair by homologous recombination. Homologous recombination repair mechanism is post-replicative repair process. When DNA damage includes a longer stretch of nucleotides, an alternative pathway is found to operate. It is called homologous recombination. DNA polymerase stops replication on reaching the damaged site in the template strand. DNA replication is resumed at the next priming site on the template strand in the form of Okazaki fragment with new RNA primer. The gap left in the daughter strand or newly formed strand may be of several hundred nucleotides. This gap is then repaired by homologous DNA recombination with the complementary parental strand using specific recombination enzymes. Enzymes DNA polymerase and DNA ligase repair the resulting gap in the complementary parental strand.